Hi everyone, welcome to Bhutan e-learning program. I am Lucky Pradhan from Brook School. Today I have come with an interesting topic, and to begin with I have few questions. The first question is, did you ever realize that we all have different fingerprints? Like you can compare it with anyone, the person sitting near to you, your parents, your siblings, or even your friends, and you will see that your fingerprints are different from any one of them. And this is one of our identities. Now, next is, though we have different fingerprints, have you realized that what makes these fingerprints unique? There should be something in our body that determines the difference or the uniqueness in the DNA pattern or the fingerprints that we have. And as I have told you, the differences in the fingerprints in us is determined merely at the genetic level, at the molecular level, by the arrangement of DNA in us. Now, how do we find that differences at the genetic level? We cannot see DNA through our eyes. We cannot separate the DNA in us, and we cannot also see the DNA that is present in our parents or in our siblings. What we can see is just the fingerprint patterns, and that are unique. And that is also being used in identification of many things, like in our olden days, we used to have our fingerprints as a substitute for our signature. Now, to proceed with what makes the DNA fingerprints looks different, or how do we actually identify that differences? And to proceed with that, I have the topic, and this is the mere focus for today, and that topic is DNA fingerprinting. Today we are going to learn about DNA fingerprinting, and by the end of this session, I would like all of us to know the three objectives that are, first of all, the principles of DNA fingerprinting. The second one is we should be able to, or we will be at least able to, describe the procedures involved in DNA fingerprinting. And finally, why do we learn something, especially in biology? We learn something just to get some applications, and we learn that topic to apply that in our day-to-day lives. Now, to proceed with, DNA fingerprinting was invented or discovered by a British scientist known as Sir Alex Jeffreys in 1984. Thanks to this man, he has been a boon to our life, that this technique helps us solve lots of issues, especially the issues related to crime, the issues that can help us solve the paternity issues, like it can unite the departed parents and children. Now, next is understanding what is DNA fingerprinting and who has discovered this technique. We have to understand why is DNA fingerprinting possible, or what makes DNA fingerprinting possible in us. And to know this, there are several principles of DNA fingerprinting that will enable us to know how it is possible to carry out, or how is it possible to identify that DNA fingerprinting is different in an individual. To go with the principles, first thing you have to know that as a biology student, as a living being, we are made up of cells. Each cell has DNA, that's also known as genetic material. Of the 100% of DNA that one has, 99% of it matches with the other person, which means 99.9% of your DNA is matching with the person immediately next to you. But what is remaining? 0.1 of the 100% is remaining. And what is making us unique? Why are our fingerprints unique? It is determined by that 0.1% of the differences in DNA between different people. Now, that 0.1% of DNA, as I told you, makes us unique. Now, how is it possible again? This 0.1% DNA is made up of different tandem repeats. These are short fragments of DNA which is inside that 0.1% of the total genome of a person. And that 0.1% of DNA has that tandem repeats, meaning short base pairs. They keep on repeating in an individual. That tandem repeats are known as repetitive DNA or satellite DNA. As I'll show you, the difference between the repetitive DNA. Repetitive DNA is of two types. There are short tandem repeats and variable number tandem repeats. The short tandem repeats, they are made up of short base pairs and they keep on repeating in an individual. It can be of 3 repeats, 4 repeats, 5 repeats, but the number of base pairs in that particular repeat remains same. Next is variable number tandem repeats, also abbreviated as VNTRS. It has, now as the term itself as variable, the base pairs can be of different numbers. Some can have 4, some can have more, some can have 80 in a person's genome, and it keeps on repeating. And next is, now still we are having the question, what makes us look different? The DNA fingerprinting involves identifying that tandem repeat in one individual. Only 0.1% of the total genome makes us unique. Having understood the types of repetitive DNA, still we are not at the clue that what makes us look different. A person who is associated with DNA fingerprinting technique has to understand, find out, or read or study the number of repeats that is found in an individual's genome. As a result, the number of the repeats or the repetitive DNA will be different. That differences will make us unique from one another. Okay, now having understood that some of the principles of our DNA, the makeup of the DNA that makes us look different, we should also understand some of the procedures that are being carried out to understand the technique of DNA fingerprinting. Now, to understand the procedure, first of all, the DNA fingerprinting is carried out at laboratory. And then next is the radio-labeled variable number tandem repeats are kept as prop. Now, you might be wondering, what is a prop? Prop is just a sample. It is made up of small piece of DNA, which is radio-labeled. And when we are actually carrying out the process of DNA fingerprinting, this prop is just used to hybridize or match the newly extracted DNA, and then it will determine what type of DNA should be reflected or what are the patterns of DNA that can be reflected in the X-rays. And it's used by, it is done by using these radio-labeled tandem repeats. Now next is to understand the procedures. We have several steps. First of all, to carry out the DNA fingerprinting, the first step is we are going to extract the DNA from a human sample. Sample can be anything. It can be a piece of hair, a piece of skin, or blood. And usually, when we extract the sample from blood, we are supposed to extract it from WBC and not RBC, which means your RBC does not contain nucleus. And you all know that your DNA is present in your nucleus. So to understand that, you have to be a little tricky, that you can only extract your DNA from WBC, white blood cells, and not RBC. As a matter of fact, that your RBC lacks your nucleus. Now next is the first step, as I have told you, extraction of DNA from the human sample. And the next thing is, in the second step, after extracting that piece of DNA, we have to see what is it like. Is it very small, negligible in size, or is it bigger? In case your DNA, the extracted DNA is very, very small, then we amplify. We make the piece of DNA a little bit larger so that it becomes possible for us to carry out the process further. So to amplify, to make the small piece of DNA into larger one, we follow a technique known as polymerase chain reaction, and through this reaction, the DNA will be added in length and it becomes a little bigger. Now in a third process, after making your DNA larger, amplifying it, the third is now that DNA is fragmented. It is digested into variable lengths using the biological scissor. Now when I'm talking about biological scissors, we usually talk about enzymes. And the name of the enzyme used in this particular process is endonuclease enzyme. And this restriction endonuclease enzyme will cut the DNA into pieces. and to tell you that these pieces are of variable lengths. They are not the same. Now in the fourth step, what we do is that after this DNA is being cut into different fragments using the biological scissor, that's the enzyme restriction endonuclease, the pieces of DNA are now loaded into the gel electrophoresis. Gel electrophoresis is a small device which has agarose gel. Here, this device is connected with electric currents, and one thing that you should know, your DNA is negatively charged. Now, since the DNA is negatively charged, it should move towards the positive end. And you might be wondering that DNA should be loaded on the negatively charged end of the electrophoresis device. Now what happens is that once you load your DNA in a gel electrophoresis, upon switching on the current, the pieces of DNA start moving from the negatively charged end of the device to the positively charged end. So here movement of the DNA fragments, they occur in lengthwise. As you know, shorter the length, faster the movement. Longer the length, slower is its movement. Now in the next step, after doing the gel electrophoresis process, the next step is that again we are going to denature the DNA. By knowing the fact that human DNA is helically coiled, it contains 2 helices. Now these 2 helices are joined together by hydrogen bonds. Now when the hydrogen bonds are being broken, the 2 helices of a DNA, they separate. Now this separation of the 2 helices of DNA is known as denaturation of DNA. In this process known as DNA fingerprinting, we use alkali to denature the DNA. Alkali usually breaks the hydrogen bond without destroying the DNA. So in the 5th process, the DNA has to be denatured. Now after denaturation, what we do is that these fragments of DNA, now we will transfer it to the a cloth piece, that a nylon cloth piece. We will just float the cloth piece over the gel electrophoresis device, and these pieces of DNA will be attached to the nylon piece. Now this is just a transferring. Now after transferring, what we do is that we fix these transferred DNA from the gel electrophoresis device to the piece, and we fix it to radioactive or UV light just to make possible for the DNA fragments to be fixed well to the nylon piece. Then after fixing it, now I had told you in the earlier sentences that we use a probe. That probe is radio labeled. Now what we do is, you know in this process that your DNA from the electrophoresis device has been fixed to a piece of nylon sheet. Then this nylon sheet has to be hybridized with the radio-labeled DNA. Hybridized means the DNA that you have copied from the electrophoresis gel, now you're going to mix with the radio-labeled one. Now after you fix it with the radio-labeled one, now what happens is that the hybridized DNA has to be fixed or let it go through X-rays. Now if you let them go through X-rays, then after that there will be dark bands of variable length observed. Now these dark bands are actually the DNA fingerprinting. Now that comparing those dark bands to the other already conducted, we'll see how you are unique from other or how that sample matches with of yours. These are the different processes of DNA fingerprinting. I hope you all have understood. Now having understood the processes, now we should see where do we apply them. How are they important to us in our everyday life? If not to us, maybe to others. But in what are the fields that are using this technique in almost every day? Now let us see some of its applications. First of all, the paternity testing. Now, as I have already mentioned to you that this technique, DNA fingerprinting, helps the departed children to unite with their parents. Not only that, sometimes those unfortunate things happen that a father of the child might not be wanting to own the fathership or maybe neglecting the child. That time, this DNA testing can be done so that we can easily identify who is the actual father of that child. The next is criminal identification, and in forensics, you might have watched lots of movies or series related to crime activities, whereby in our daily lives also it has become really important that we have important interesting technique, easy technique that will help us identify the criminals. Otherwise, other technologies might write off that who were the actual criminals. So this technique, DNA fingerprinting, has been very interesting or helpful to us, that they make us identify the criminals easily. If a crime scene has happened, and if we need to identify the criminal, we can always take the extracts of DNA from something, maybe a pieces of skin or blood that the criminals might have left on the crime scene, and we can do the DNA finger testing, and we can always identify who the criminals are. Now the next application is that this DNA fingerprinting is also very useful in identifying the genetic ancestry of human population. It means that we have our ancestors and we don't know how they were, and we also don't know when they died. But sometimes the scientists, what they do is they perform, when they find out the fossils of a dead human being, they perform this genetic fingerprinting to identify the nature of our ancestors. And also through this, we will be able to understand how they differed from us. Now next application is the fourth one. This DNA fingerprinting is also very much useful in studying endangered species in conservation biology, which means here we are talking lots of, or giving the ideas about conservation. It can be conservation of plants, it can be conservation of different species of animals. So in biodiversity, to conserve the endangered species, the species of plants and animals, they are likely to be vanished. We can always study about their genetic diversity by studying the genetic fingerprinting in them, and we can always look forward for conserving them in better ways. The next one is gender and identity of deceased person. When I talk about that, sometimes in natural calamities, what happens that a person dies, and when the person dies, it happens so that it will be difficult to identify who that person was. What was the age of that person? What was the gender of that person? And whom that person belonged to? Through this DNA fingerprinting also makes it possible to identify the gender, whether that deceased person was male or a female, and whose child was that person, or also who was that person, what was the age of the person. These can be done. Now these are the applications of DNA fingerprinting. Now next is, now I would like to summarize the topic. To summarize the topic, I'd like to take you through the video. Please watch this short video. This summarizes the entire content.
My house was broken into last week. The police can identify the culprit from blood found at the scene. How can they do that? To understand this, we need to state what is meant by DNA fingerprinting and describe some uses of DNA fingerprinting. The burglar smashed a window, leaving blood on the broken glass. Samples of blood, hair or skin can be collected from a crime scene. Forensic scientists investigate the samples in a lab. First, they extract DNA from the cells. They use enzymes to cut the DNA into shorter fragments. The fragments are placed into a well in a gel. They are separated using an electric current. Different sized fragments travel different distances in the gel. The smallest fragments travel the furthest. A pattern of bands is produced. This can be compared with the patterns of DNA from possible suspects. The enzymes cut different people's DNA in different places, producing a different set of fragments. The different sets of DNA fragments will produce different patterns of bands when separated on a gel. Each person has unique DNA, which will produce a unique pattern that can be used to identify them. If two patterns match, the samples are from the same person. So, the police are able to identify the man who burgled my house using his unique DNA fingerprint. They make a DNA fingerprint by taking a sample of cells from someone and separating fragments of DNA to make a pattern. If the pattern matches a sample from a crime scene, that person was there.
I hope now, after watching this short video, you have been able to summarize whatever content we have learned so far. Before I go, I would just like to bring about the differences between the conventional fingerprinting method and DNA fingerprinting. As you can see that the conventional fingerprinting, it was widely used in olden days. Now it is quite less used now, and then it is being overshadowed by this new technique known as DNA fingerprinting. This is because of the fact that the fingerprinting method, they were actually, since we were identified with the fingerprints of us with that of others, now what happened was that because of technology, people could rub or change their fingerprints by plastic surgeries. So because of that, identifying a person with the help of fingerprints made it difficult for an individual. But now here, this new technique, DNA fingerprinting, it is difficult for an individual to do any surgery because these changes come from within, from the genome. So the differences remain forever. Now to conclude with today's session, I have takeaway questions for you. First of all is, this question is simply the question that you have to summarize from the content. Now I'd just like to read the question for you. Here I begin. There is a paternity dispute for a child. Which technique can solve the problem? Discuss the principle involved. Now here, you just have to discuss the principle and process that is involved in DNA fingerprinting. Now next is this question. It's a thinking question for you. Can identical twins have a same DNA fingerprint? Why or why not? So please think over this. So thank you. Hope you all have enjoyed this session. See you again.